I'm making this recording to answer a question during finals week concerning the difference between changing the frequency or the amplitude of nerve stimulation using an electrode. This is similar to what we did in our finger twitch lab early in the semester. First, we have to define a few things. If this cylinder represents our forearm, then the strands running inside this cylinder are now representing a nerve, which is a bundle of neurons, and each individual strand represents a single neuron. If we then activate our stimulator, it will send an electrical signal from the positive to the negative terminal of the stimulator, and that electrical signal will also pass through the tissue and have the potential of activating motor neurons that it encounters as it goes through the tissue. Every time we activate the stimulator, we activate that same population of neurons. If we increase the frequency, then what we see is that we get greater or more frequent responses from the motor neurons, which leads to more frequent responses from the muscle cells. And you can go from a single stimulus, which produces a twitch, to very fast stimulation, which can produce tetanus, but only for those muscle cells that are innervated by the motor neurons, which are reached by this electrical signal. In order to recruit more motor neurons and to then get a larger muscle response for a larger population, we increase the amplitude or voltage of the signal. When you increase the voltage of the signal, then you can sample a larger population of neurons, and then they will respond. And if you give a single pulse, you'll get a twitch. If you increase the frequency of the pulses, then you can eventually go to a high frequency and get a tetanus, now of a larger population of muscle cells, because the electrical signal activated a larger population of motor neurons. I hope that this makes it clear.